Hi everyone. We don't need to Thanks. Hi everyone. Thanks very much for joining us at this morning's SAIAB seminar. Just some house rules before we begin. If you've got phones on you, please make sure they're on silent. For those joining us online, please can you keep yourself muted at all times? And if you have any questions throughout the talk, please can you put them in either the chat function or the Q&A tool. With that, it gives me great pleasure to introduce Dinah Mukhari and Bantony Ziko, two of my PhD students who are both in their third year of their studies, and they've been very hard at work learning all kinds of cool things about mullet. Today, they're going to be telling us a bit about their recent Otolith microchemistry work and a very cool 3-week trip that they took to France. Looking forward to the presentation. So with that, over to you guys.
Okay, good morning, everyone. Thanks for the introduction, Taryn. And before we dive into today's presentation, I just want to take a moment and thank my wonderful 2 supervisors right here for making this project possible, and our collaborators in France who helped us with organizing everything from our paperwork to the lab when we were there. This is one of those projects when you write about on an abstract or on a proposal, you never really think it will happen. You never really think you get this opportunity. But when we finally did, so I'm looking forward to sharing this and with my colleague Bantony, who will continue after. So, sorry, can I just say from the title, From Chemistry to Conservation, using Otolith to track fish movements. So what we do most of the time in ATAP is use acoustic telemetry to track fish movements, but we're going to take you through a different method that we also going to have as part of our PhD chapter, which is using microchemistry. So this is the chemistry part, but we're going to try and break it down to be understandable. So please don't sleep. Just to get into our study species, which are mullets. These are small fish that are dominating estuaries across the world. And then they are known to spawn at sea. I have a life cycle here, which shows them as adults, they go to sea and spawn, and then go back into the estuary, and sometimes even go into the river. So they move between the sea, the estuary, and the river throughout their life cycle. And then we want to find out where they spend most of their time and why are they in certain habitats at certain times. So mullets are also important economically and ecologically as they are food sources. They are harvested in some countries as primary food source, and then they are also used by recreational fishers as bait. Here I have a picture of somebody using mullet to catch bigger fish like shark. And there's also lack of movement information, especially in the Southern Africa, despite some of the species even facing overexploitation. These are the 2 species that we focused on. So the groovy mullet, this is their distribution. It's mostly in Africa. And then this is the flathead mullet, which has a cosmopolitan distribution, which is found across the world. They mature at different lengths. So groovy are a bit smaller than flatheads. So they are 180, and the flatheads are quite big. And then they grow up to different sizes. It's interesting to see that the flathead can get close to 1 meter. And they have different spawning seasons as well. And these studies were done in KZN. And then the aim of this study or these chapters were to determine the habitat use pattern and connectivity between habitats. Habitats, I'm referring to the sea, the estuary, and the river throughout their life. So there's multiple methods that you can use to study fish movements. Mark and recapture, whereby you catch fish and then you mark it, and then you recapture it later at a different place, and then you can say this fish moved from one place to another. And then we have satellites, we have acoustic telemetry, which is what ATAP does. And then we also have eDNA, and on the recent decades, the development of Otolith microchemistry. So this is our study site, which is in Kawi in Port Alfred. This is somewhere there in South Africa. And then at the Kawi, this is an estuary that starts from the riverside and goes down all the way to the mouth here. Most people have been around this area. So this is the lower reaches of the estuary. And then they have different salinities. If you compare the lower, the middle, and the upper being a bit freshwater based, and then middle being somewhere in the middle, and the lower being high in salinity. And this is going to be important for later. These are the methods we use to catch the fish. So just before we started this project, we received an online training by Sea Unicorn on how to use Otolith microchemistry. And then we use different methods to catch the fish. This is seine netting and cast nets to catch mullets. And to say it was a nice experience, not really. We struggled to catch the fish, but when we eventually did, it was an exciting part of the project because we used this fish for multiple reasons. So we started by using this fish for thermal tolerance experiments. So we took this fish down to the marine lab at DIFS, and then we ran some experiments on them to determine their thermal limits, temperature in terms of maximum temperature that they can tolerate, and the minimum temperature that they can tolerate. And then to know which fish is which, we had to at least put some kind of tracking on them or to mark them. So we use some dye to mark the fish, and different dyes will be associated with an individual fish. So we can keep track of which fish is which, even continuing later to the otolith extraction. So we took out the otoliths from the fish after the experiments were complete, and then we made sure to store them in dry containers in preparation for this particular project. So this is what an otolith looks like on a closer view. This is not of a mullet, but this is what it will look like for most fish, although they're different in terms of size. Yeah, so in a fish, otolith is for hearing and balancing is on the ear of a fish. And then in research, we use it to know the age of a fish. So just like a tree and the rings that can tell you the years of a tree, how old the tree is. With the otolith, we can do the same thing about a fish and know how old a fish is based on the rings on the otolith. And then we can also do growth and life history analysis. Look at the rate of growth. Is it growing faster at a certain season compared to another season? And then we can do migration and habitat tracking, which I will explain later, and environmental monitoring. So what happens on otoliths is that every day of a fish, a certain chemicals are deposited on the otolith. So if you take a fish that's 3 years old, you'll be able to see where that fish has been in that time. And then every single day there will be deposits, and then over a year. So what happens during winter, they don't grow as big as they do in summer. So this would probably represent 1 year, and then summer growth, another winter growth, second year, and going forward. And then as they grow bigger, their growth start being lesser distance apart compared to when they're smaller. So microchemistry is looking at those chemicals that are deposited on an otolith to see where or which environment was this fish at from the time of birth to the time of capture. So as I mentioned, where we caught the fish, so there's different salinities between the sea and all the way to the freshwater. So we are planning, we were hoping to find the difference being shown on the otolith as we go from birth, which I mentioned that most of them are born at sea, so you can see some higher elements there. And then they move to the estuaries. Some of them return to sea, some of them don't, or go in between over time. So that's the information that we wanted to see on our fish. So the variation in environmental condition changes due to chemical composition of the environment. And then we can measure those differences and infer something about the individual's past location or history. So this is the chemistry part of it. But so there's about 50 elements that you can detect in an otolith, and then 7 of them can be used to infer past location. So 7 of them can be used to say where this fish was, whereas the other ones can be in low quantities and you can't use them. And then most marine fishes have about 83 to 98% of their otoliths is made of strontium and barium, which we will talk about in this talk. And then we usually use strontium to calcium ratio or barium to calcium ratio to look at where the fish was. So if strontium is positively correlated to salinity, which means that environment where there's high salinity, there's usually high strontium. So in the sea, that's when you find high strontium. And then barium is negative. So at the sea, there'll be low barium, and in the freshwater, there'll be higher barium. So those are the patterns that we want to see and infer the locations. So I'll leave the presentation here for my colleague Bantony to take you through our tryptophans.
Thank you so much, Dinah, and thank you everyone for coming through today's talk. Yeah, I hope you are still around for what Dinah was explaining. It's only gonna get easier from here. So I hope that you understood what she was talking about. Yeah, just to continue on what Dinah was saying. So after we collected and extracted our otoliths, we had to take them to the University of Montpellier in France. And we're in that building, like for all the 3 weeks that we're there, we just, they're busy in that lab for the whole 3 weeks. So I'll just take you through what we were doing there. So the first thing that, so we had our otolith samples, and then, okay, let me just put a pointer. Okay, so the pointer. Okay, so after we have our otoliths extracted, so we had to put them in a mold which contained a resin. So here's the mold, then you put a resin, and you allow the otoliths to set and then to dry. And once you do that, once it's all set and dry, the next stage was to locate where the core area of the otoliths was. So if you look at this image here, the picture of the otolith, so in the middle here, that's where you find the core or the nucleus, or let's just call it the center of the otolith here. And once you identify it, you have to make a cut because you don't need the rest. You just need to focus on the core of the otolith. So if you see this yellow line here, it's where the core is, and then we wanted to make a cut through these lines. So you put a line through the middle of this line to identify where the core is, and using this magnifying microscope, you make the line to indicate where that line is. And after that, you go to this part here, which is just a blade to cut the otolith. So you're gonna make 2 cuts, and then you'll be left with just the middle here. So yeah, so that's that for this part. And the next thing, yeah, that photo is completely irrelevant for me. Like, that should be there. Okay, so once you make your cut here, so this is the cut that you make. So the next stage was to polish the otoliths. So it goes through a series of papers that you will be polishing to make it clearer, and also to get to where the core of the otolith is. So you use different kinds of polishing paper that have different grades. Some are much coarser, some are smoother than others, until, so each and every time you polish, you have to look if you have identified or getting closer to the core of the otolith. And so this is under a microscope. If it's not okay, you can go back and then polish again until something is clear. And then the result will be something like this, what you see on these 3 otoliths. So for these fishes, they are different sizes, different shapes. they're never the same. So you have to like treat each otolith as unique. And then once you have this, the one in the middle you can see is more or less clearer than others, but you can see here at the center, that's where you find the core of the otolith. And then, so this dark area usually represents the larval life of the fish. And then as it continues to grow, you get the iron rings, which you get the first iron ring and then the second. So in terms of aging, you would assume this is probably a 2-year-old fish. And then after you polish and get these, the picture, this room behind me, you're supposed to, when you enter it, you're supposed to enter it wearing this type of clothing. So you're supposed to cover your head, your feet, and all of that. And as I said, it's not relevant. And then once you do that, you're going to put your otoliths that you have polished on a slide. So you're going to tap them here. I know the picture is not very clear, but these are otolith samples that are on this slide. So you have different samples, and then after that, this is what you're going to take for analysis on the machine that we actually went there to see and to use for our studies. So the machine that we use is called a laser ablation inductively coupled plasma mass spectrometry. Okay, you can read for yourself. Okay, so yeah, once you get your otoliths and put them on a slide, it goes through this machine, which I'll just briefly explain. So the slide. So the slide which has your otolith samples, it goes and then it goes in here. So how the sort of like this machine works, so there is two components. So there is a laser component and then there's a mass spectrometry component. So once you put your samples, so the laser is going to, so this is just a beam of light, or usually about 1 micron, is going to ablate your sample that you put. So it's gonna go through each sample that you have here. And then once that, then this argon, or it can be argon or helium, that's the one that's going to be released into the sample. And then that will cause the ionization process where the ablated material is going to be ionized, and then after that, so it changes into charged particles and then goes into this ISP, and then they are separated according to their mass and charge after they've been ablated. They've been ablated through the argon or helium gas, depending which one is used. And then, so because there are different elements that you get from the ablation, so they are separated according to their mass and charge. And then that's how they are separated. So if you have, let's say, an element like calcium, it's different from an element like magnesium. So they're going to be separated based on that. And then once you have that, so that's all happening in the background. But what we will see in the computer room is you first of all have to select the elements that you need to analyze. So here on the first slide here, you just select elements on the periodic table that you want to analyze. And then here is what in the middle of the video shows what's actually happening as the laser is going through your sample. So you have your slide in the laser machine, and then now the laser is going through the otolith. So each time it's going to make a spot along the otolith until it reaches the end of the otolith. So you start from the core of the otolith until you reach the end. So it's making spots, many spots until it reaches the end of the otolith. And each spot is going to have different chemical elemental compositions. And then once you have that, you can still visualize that while this is going on. So you can select, let's say, an element like calcium when it starts, and then you get different peaks. So it shows you how it's changing on every spot that as the laser is moving along the otolith. I don't know if you are still together. Okay, cool. The fancy stuff is over now. Yeah, so for our preliminary results, we are still analyzing most of the samples because we still need to filter a lot of data and cancel out a lot of things that are not important. But what we have here, we have barium and strontium concentration. So as Dinah explained, that we usually use barium and strontium because those are the two elements that kind of vary between the sea and the estuary. So you want to see if there are any changes between those concentration of those elements. So for example, here, so these lines on the x-axis here, it represents the number of spots that the laser was taking from the core of the otolith. So this is the first spot that the laser was making. So you can see it is up to 241. And then on each spot, it's going to measure the concentration of barium in this case. So these lines are lines for different otoliths of different fish, right? And then you can see how the concentration of barium is changing throughout the number of spots, which is the distance from the core. So since our fish, let's say if you take the blue line, started here and then increased a bit, then it stayed a little bit constant, and then there was a change. So you can sort of now or predict or assume that, okay, something changed from this point to here. So let's say you can assume, let's say in the sea, that the barium was low, and then maybe here the fish entered into the estuary, and then there is now a peak for barium. And then you can have another fish doing something that is completely different. And also because these are estuaries, so there's going to be a lot of fluctuations with that as well. And then we also measured the strontium concentration, but I think you're as confused as me. This is a lot of things going on. Can't take away, or at least not now, but later on. And then, so here we actually have, these are results from some of the fish that were juvenile fish. But here it shows, I think, a better story than the previous one. So here you have, starting from the sea, then you have sort of these similar peaks, and then at the end, it sort of drops down. So remember when some of the fish were also kept in the lab for some time. So we are predicting, or the prediction is that at sea, since they were kept in the lab with using seawater, which was the salinity is pretty much the same as the one at sea, which is usually where they begin their life at sea. So you can see that it is almost the same during the start and then at the end of their life, which is where when we kill them. Yeah. So these are kind of similar concentrations. So at the beginning of the larval life here, and then this represents when they entered into the estuary, and then when we captured them, we kept them in the lab with conditions that are similar to the sea. So that's sort of information that we are looking at. And in other studies on the diagram on the right, so they can also show this strontium. For for this particular study, they were using strontium, and then it shows different peaks. So you can have strontium increasing and then decreasing. So from that, you can infer which habitat that the fish was in. So if there are fish that migrate between the freshwater, the estuary, and the sea, you can tell from those different concentration of your strontium where that fish has been. So you can tell whether it's been in the freshwater or in the estuary or at sea. But you still need to validate those concentrations as well. So we're still working on that, and I think we'll present the results later on as we find them. And I think this is the exciting part. Yeah, Dinah, you can tell them the exciting part. But anyway, we were at the University of Montpellier, but we also got on one or two occasions also got to visit the nearby cities. So we're in Montpellier, and then we also got to visit other cities that are nearby. So there's Set, which is a coastal city with a lot of, you can see the boats that we don't have such boats in South Africa. They have a lot of sailing boats. Well, that's what I'm trying to say. And then, of course, yeah, this is the library in Montpellier and the famous Eiffel Tower. This is in Set, yeah, just a lighthouse. And I don't know what this thing is, but it's very old. So yeah, I think we're ready for questions, right? Yeah, I think with that, we would like to thank everyone for coming through to listen, and everyone who helped in catching the mallets, which were difficult at first. And also want to acknowledge all the collaborators for this project, our supervisors, Ian Bantary. Thank you so much for this opportunity. And yeah, I think that's it.
Cool, thanks very much. That was amazing. Okay, questions? Interesting.
Merci. Very, I love this Otolith work for connectivity. One question that I might just go beyond your scope, but you targeted the most charismatic elements, basically, that say it's going to be marine or freshwater. Have you considered also to maybe look at some water samples that could characterize and in a way validate the chemistry of the water in the sea and freshwater, sometimes also enabling you to look at, if you've got the telemetry data, so enabling you to further find relationships with smaller temporal scale?
Yeah, okay. So for validation, we were looking at, since we kept the fish for extra time in the lab, and then we know the conditions of the lab because we recorded them. So we wanted to compare that with the ones we find on the Otolith. But then when we were analyzing the data, we do realize some spikes in different elements actually. And the next step is to also see why, like, because yeah, strontium and barium, I expected to do that, but magnesium, for example, we didn't expect the kind of patterns we were seeing. And then there was also, what is it? lead as well, could be contamination, but we're also exploring to see what that could be.
Kawi is also an urban site towards the mouth, so you could pick up elements from urbanization, micro elements from there.
Yeah, I think we even considered having some water from the to validate our results for them.
Thank you.
Yeah, I don't think that, sorry, I was really great talk, and it's awesome opportunity you guys had. And you guys obviously focusing on strontium and calcium and beryllium, but going on to following on from that question, I mean, you know, I don't know if otoliths have been used as sort of ecological indicators. So given you did in the Kawi River, which is quite urbanized, etc. We know a lot of our estuaries are under a lot of threats. And this is obviously not the scope of your project, but would it be possible, for example, another student to look at the heavy metals that are of concern, lead, you know, the other mercury in these, you know, in these mullet otoliths, to see whether the time that they're in the estuary are they exposed to heavy metals. You know, most of the other heavy metal analysis I've seen done have been mostly like in flesh. So when people have sampled fish from a river and then looked at what's happening there, but you could potentially, with the data you have, sort of track where if there are spikes in. Yeah, it's just I think the data set has a lot more to it potentially than just movement, if you want to look at it differently. But yeah, well done.
Yeah, awesome talk, and I'm really jealous that you had a way more fun than me. But I'm just curious, how does temperature affect the uptake of barium and strontium? So if it's warmer in the river and obviously the otoliths are growing a bit quicker, does it take in more of these plant trace elements? Or does it concentrate? How does it work?
Yeah, so yeah, usually temperature does. So in terms of, I think the main two variables, salinity and temperature, are the ones that usually affect, or especially the elements that we are looking at, barium and strontium. So it kind of affects those. I'm not sure about the other elements, but yeah, generally, like there's an increase in the uptake of those elements when the temperature increases, for example. Yeah.
Just to add on our validation, which is still happening. So the fish that were in the lab, especially the juveniles, because we can see more on a juvenile than an adult. So the fish that were in the lab, we know the temperature that we kept them on in the lab. And then we want to compare if that growth they have on the lab signature will be similar to the beginning of their life, or is it gonna be too different? So in the lab, I think it was 18 degrees. And then we know the sea temperature from the weather data. And then we're gonna compare if the intake of barium or the strontium, does it change a lot or a bit less? Yeah.
Any other questions in the room? Yeah, Makamaka.
Yes, very interesting talk and exciting. The big project was reading my mind. It would be very nice also to see if you could compare the other sources as well. So Dinah showed a very interesting slide saying there are 7 elements used to assess the age.
Yes.
The age, yes.
Found on Otolith.
Yes,
Yes.
and then the Bantony shows a cool slide about using of ICP spec, which is something that I like the most. Do you have certain range of concentration that you can judge if it's, for an example, if you have 50 milligram or microgram of cadmium and 5 milligram of cadmium and whatever, that means this species is old or it's young. How do you then judge?
For age.
Yes, based on you base the judgment of the concentration values?
I'm not really sure about using that for age, but for age, we usually look at the rings.
The rings on.
Yeah, on the otolith. And even the size of the otolith is quite different. So I'm not able to go back here.
I mean, it's fine.
But so on the like this one, for example, this would be the larval life. So before 1 year, and then there will be 1, 2 years, 3 years.
Okay.
Yeah, but I don't know about using elements to look at age without the rings. I'm not 100% sure. Amber.
Yeah, just out of interest, we don't normally use the elements to correlate with age, but when we did COG, we found that magnesium spiked at every annually, so we could actually use magnesium the spikes in magnesium to look at the age, and then nothing was available at the time, but subsequent to that, there have been 1 or 2 papers which have related magnesium to somatic growth. But yeah, I'm not certain. don't know if you
What's that?
Is it possible maybe for someone else to do similar study to study the daily movement of fish, because there might be those that are moving in between, since you said that they entered the estuary during larval stages.
Well, daily, I think that's quite challenging because of I think the resolution won't, yeah. I think you have to be like so precise, but we do have other methods like acoustic telemetry that can do, that can do daily movements. Yeah.
Just to add, can I
Yeah.
add to that? So if you're doing daily, it will also depend on the speed in which they move. Let's say they go to sea and come back so fast, how sure are we that it's gonna be recorded on the Otolith at that speed? Yeah.
Okay,
So the question relates to ontology and the movement patterns, like in terms of, is there a possibility of maybe confusing the spike and say this is due to environmental conditions while it's due to the growth and the deposit of these trace elements into the Otolith? And if that's the case, is there a way of maybe filtering through, getting rid of the changes or the patterns that you see that are due to growth?
We haven't confused it yet, but one thing we've noticed is that at the start of life, because at the core is where the life starts. So at the start, there's usually a high barium concentration, like this one, for example. So at the start of life, because when I first saw this, I was like, why is this one high at the beginning? And then Audrey, who was helping us that side, was like, this is probably because you started exactly at the core. And then this one could be that you started maybe one day after. So the barium is high at the core, that's what we know. But going forward, we haven't seen it being too high to be confusing.
Okay, any questions in the room? I'm going to jump back to you now. Let's just go online. Alan says, did the preliminary Otolith analyses show any habitat use differences between the two mullet species?
Thanks, Alan. Yeah, we haven't really gotten to that part yet. This part we're still analyzing individually to check, to actually, first of all, validate if we can use barium and strontium as good signatures. And then after that, if it makes sense, then we're going to compare between the two species. Yeah.
So I think the joy of this whole study is that it's forming one thesis, one chapter of Dinah and Bantony's thesis. So there's so much more that they could go into, but it might be a thesis on its own. So, and then I think there was one more question from Dumi. Can this method be site specific? Also, is it possible that your validation could be influenced by some environmental change like flooding?
First, thanks for the question, Dumi. Site specific, I don't think so. Unless if I
Yeah,
don't,
it will be.
it'll be.
What is it?
So it's like estuaries, between estuaries. and
Oh,
stuff.
Oh, okay.
Oh, yeah.
And flooding, what are your thoughts on flooding?
Yeah, of course. It will potentially, but I think it depends how long the flooding also occurs, because it has to allow enough time for the elements to be deposited on the Otolith so you can pick it up. So if it's just a few days of flooding, you might not pick it up. But if it's like extended, then obviously there's a lot of fresh water, so barium potentially would increase. So
And that's assuming the fish stay on.
yeah, also assuming the fish stay in one place. Yeah.
Okay, Roger?
Yeah, how sure are you that you're getting annual growth rates right down? I mean, years ago, I used to open those studies where they were validating the year. I could imagine that, moving from a marine to a freshwater environment, you've got variable growth rate. And is that a standard that you pursue, you establish that?
The first part of the question I didn't get, how sure are we that?
The growth rings that have been laid down, the counting with annual rings, are they actually annual rings?
Oh, okay, okay, that's a good
They
question.
established that they are?
Other age and growth studies.
Yes.
Yeah, that's been done.
So, yeah, so usually the age and growth studies do the validation beforehand, but our study is not really too much about the growth and how old the fish is. So it's not the primary focus. So we just want to see their movement over their lifetime instead of how old were they when they went to the estuary and when did they go to the freshwater, because they do go up and down a lot. So we just want to see if there's any potential impact or any site that they prefer during their lifetime in general. And then we have, because we took out 2 otoliths, so we have 1 otolith that we use for this, and then the other one that's still untouched. So should we need to do age and growth, we'll use the other one that is not polished yet and that we didn't affect in any way. But we haven't done any validation so far.
Any other questions? Francesca?
Basically, now you've got the telemetry chapter and this chapter, you're gonna validate against one another to see if your telemetry or if your otolith's results match and or which method do you think is better for what scales, what life stages or something like that.
Yeah. Yeah, we were going to to sort of combine the two. So this they both give us like the movement that we want, but also I think with telemetry it's going to give us like you're going to have like a sort of a real time movements from where you caught the fish, and then these ones you're going back in time, and then telemetry you're going there. So with the patterns that we have for the telemetry, which we've already analyzed the data, yeah, we can kind of see with the adults, I guess, the patterns of up and down the estuary. And so, yeah, it's a lot of movements. That's what I can say for now. Yeah.
Last question, sorry. Do you have any otoliths from your fish that you tagged that were recaptured?
No, we didn't
You don't
recapture.
have any?
It would have been so nice to recapture the same fish and use them for this.
Doesn't matter, even if the tag's dead, then you can still look at the track and then look at that part of the otolith. That would be good validation.
Yeah,
Yeah.
it would be nice to do that, but having this opportunity again comes once in a lifetime.
Oh, you're gonna send them, you're gonna send them.
Oh, yes, we can send them and find out. Yeah.
Yeah, when it changes in terms of concentrations of the barium across different size classes, across maybe different years, the reason why I ask this is this in line in terms of managing of this particular species?
Yeah.
Any recommendations, if there were changes in terms of maybe 10 years back, the species were moved from freshwater to marine, maybe between the years of 2 and 3. And then now, 10 years later, those changes, there were changes now. Now it's moving when it's a bit younger, between 0 and 2 years.
Okay. I think the nice part about this presentation is that it's been recorded because there's some really great developments we can do, but we haven't done them yet. So we
So I think the study is really a cool study. So I think it has huge implications in terms of managing any particular species.
Yes.
And so we are well done there.
Thank you. Yeah, I think we can find out a lot more even about the environment from this otoliths, yeah, going forward.
Okay,
Yeah, so the nice thing with the Otolith is that it's the lifetime use of the issue with the telemetry is just a snapshot, so you can actually go back and so they complement each other. But one thing I want to say, there's two types of Otolith microchemistry. So this is the laser ablation, which generally people focus on barium or strontium, which is salinity. But if you wanted to look at estuary specific, where those fish came from, if they came from specific estuaries, then you could go to this particle and actually dissolve the entire otolith, but then you need like tier 0, tier 1 fish, dissolve the otolith and get signatures, and then basically get or catch fish in different, so from the Kawi, from the Sundays, from the Swartkorps, and then look if those specific estuaries have concentrations. And then you could do like a whole multiple analysis. But that is different. So you don't actually laser through the lifetime, you just basically dissolve the otolith that's just got a year of growth from the estuary that you've collected it in. And then, yeah, that's another way, but that's a whole different analyses, and that's when you have to really not contaminate the otolith.
That's it. Collecting the otoliths, is it standard? Is it different for this particular work? Or you just take a lot, put it in the, it doesn't deteriorate through UV light or anything?
So the one that Amber is talking about, where you need to dissolve the whole otolith, you will have to be really, really clean on the collection and what you use to put the otolith in the epindorf and everything else. You need to be really clean. But then this one, you can use your standard otolith collection, although we were using plastic instead of metal pipettes and stuff.
Given you section it,
Yeah.
I mean, the middle of the section cannot be it's not contaminated by the outside. So
Yes, even
I would
though,
think maybe that only the absolute edge might be contaminated by the storage environment, but if you're sectioning, wouldn't make sense if it's inert. Because if it is contaminated from the outside to the inside, then the otolith isn't inert, and the otolith is.
No, exactly. That's why you don't need to be
Yes.
so careful with doing laser
Yes.
ablation. But the solution base is you have to use high density polyethylene tweezers. You actually have to extract the otolith in a clean room, because obviously any touch, if there's like high levels of contamination, will just destroy your sample.
Okay, I think we're gonna stop it there. Thank you very much for taking the time to listen there. Thank you.